Good afternoon, everyone. Good afternoon, everyone. I, Assistant Professor Sharya Pravin Rathod, welcome you all to the second session of the sixth day of 1 week online international e-FDP ESTP entitled Empowering Pharmacy Education, Navigating Regulations, Global Trends, and Advancing Outcome-Based Education and Research under the aegis of IQAC in association with APTI. It is my pleasure to introduce our second speaker for the day, Dr. Bothiraja, an esteemed associate professor at Goa College of Pharmacy, Panaji, Goa. Today, Sir will enlighten us with his expertise on the topic how to select the drug molecule for formulation research. Sir holds a PhD in pharmaceutics from Bharati Vidyapit University, Pune. Prior to that, he earned his postgraduate in biopharmaceutics from Goa Government College of Pharmacy, Karad, and graduated from MGR University, Chennai. Dr. Bothiraja, with over 22 years of academic experience, is currently serving as an associate professor at Department of Pharmaceutics in Goa College of Pharmacy, Goa. Sir is specialized in subjects like biopharmaceutics and pharmacokinetics and novel drug delivery systems. Sir has also worked with Pune College of Pharmacy, Pune. Dr. Bothiraja's career is marked by several prestigious accolades, including being a 5-time GATE qualifier and a mentor for the winning team of Smart India Hackathon 2020 organized by IIC Cell MHRD Delhi. His achievements include the Teacher Associateship for Research Excellence Award 2019 and the Best Researcher Award at Bharati Vidyapit University. Sir has also received Best Research Paper Award at the 4th Conventional Society of Ethnopharmacology, Zadavpur University, Kolkata in 2017. Sir has delivered 52 invited lectures globally at various institutions, national, international seminars and conferences, and has been a PhD reviewer at various universities across India. Sir's research contributions have greatly impacted the scientific community with an impressive record of 63 publications, including 54 international and 9 national papers. With 2049 citations, an impressive H index of 27, and I-10 index of 50, his work has not only contributed to the academic world but also led to the filing of one patent and the contribution of one book chapter. Dr. Bothiraja has also been the recipient of significant research and consultancy grants amounting to approximately 2 crore 93 lakhs. Among these grants, we have DST SERB TARE with a funding of 19.80 lakhs, which is currently ongoing, followed by CCRUM Ministry of Ayush Government of India grant, which supports his research with 43.57 lakhs, also an ongoing project, and the DST FIST Government of India grant, contributing 72 lakhs towards his ongoing work. Dr. Bothiraja's dedication to advancing knowledge is truly commendable, and we are fortunate to have him with us today. Let's give a warm welcome to Dr. Bothiraja, sir. Now, without further ado, I would like to request Dr. Bothiraja, sir, to please take over and start with the session. You may start your session, sir.
Yeah. Is it visible now? Hello?
Sir, you are audible. You may start your session.
Yeah, my screen is visible. My screen is visible or not? Just I'm asking.
Yes, sir, your screen is visible.
Okay. Shall we start?
Yeah, sure, sir, you can start.
Okay. Good afternoon to all of you. First of all, I would like to thank to principal as well as the coordinator of this FDP, Gosavi College of Pharmacy, Nasik, for inviting me to deliver one of my topic, how to select the drug molecules for formulation research. This is mainly useful for PG student as well as PhD scholar, those they are working in the pharmaceutical department, especially in the research and development. So my presentation will cover the three part. One is a high throughput screening. From that, we used to select the lead molecule. Selection of drug molecules, nothing but selection of suitable drug molecule from the lead molecule for formulation research. And some of the exercise, I will put some of the exercise drug property on the screen. The audience, you people should suggest what are the formulation is possible for that particular drug molecule. That from exercise to the audience. So high throughput screening, nothing but lead molecules. Lead molecule means nothing but any medicinal chemistry people used to synthesize n number of molecule. From that, which molecule have highly potent nature or potent therapeutic efficacy, we used to say it is a lead molecule. So once identify the lead molecule, then it comes to the pre-formulation scientist. Then again it comes to the formulation scientist to develop the suitable formulations. That is called as a, we can say, lead molecule. Okay, so medicinal chemistry people used to synthesize n number of molecules and they used to finalize the structure by IR, NMR, MOS, and all those things. Finally, the medicinal chemistry people used to say that this molecule have the particular therapeutic efficacy or particular potency. Like from the basic nucleus heterocyclic ring, and they will suggest that this molecule having anti-cancer activity, analgesic activity, or antipyretic activity. Like this, they used to say from the structure it has the so and so pharmaceutical or pharmacotherapeutic effect. That molecule will come to the pharmacology department. What pharmacology department used to do? They used to analyze the therapeutic efficacy as well as the toxicity. If molecule having less efficacy, more toxicity, suppose, the molecule will go back to the again medicinal chemistry department to modify the structure. Why they used to modify the structure? To increase the efficacy, to decrease the toxicity. For that purpose, they used to modify the structure. Once modify the structure, again they will confirm the structure by IR, NMR, MOS, and so on. So after that, again the molecule will come back to the pharmacology department. Again, pharmacology department will analyze the therapeutic efficacy as well as the toxicity. If it has more efficacy, less toxicity, then that molecule will be considered as a lead molecule. Once the drug has the more efficacy, less toxicity, then it comes to the free formulation scientist. They will go for the free formulation study and followed by formulation development. So whenever the medicinal chemistry people used to modify the structure of leads to increase the lipophilicity of the molecule. Whenever if you modify the structure, there is a possibility to increase the lipophilicity of the molecule. Once the molecule lipophilicity is increased, automatically it will affect the aqueous solubility. Once aqueous solubility is affected, automatically bioavailability also going to decrease. At the same time, whenever you modify the structure, there is a substitution of some amino group, hydroxyl group, carboxyl group, something we are substituting to make the high potent molecule. If we are substituting, there is a possibility to increase the molecular weight. If anything molecular weight increase, again there is a problem in the solubility as well as the permeability. As we studied in the biopharmaceutics, anything drug want to go absorption or permeability, the molecule should have the molecular weight of 100 to 400 Dalton molecule. If anything beyond the 400 Dalton molecule, like steroids molecule and all, very difficult to go for the absorption process. So once you modify the structure, there is a problem in solubility as well as the permeability. First, we'll discuss about permeability, then we'll go to the solubility. So, whenever any molecule shows the less permeability, it means what? The molecule might be having the lipophilic nature, or sorry, it might be having high molecular weight, or it may be the hydrophilic molecule. That means more hydrogen bond count present in the molecular structure. Sometimes what will happen when you modify the structure, sometimes it will convert it into lipophilicity, or sometimes it will convert it into hydrophilic molecule also. Once it is a hydrophilic molecule, again problem in the absorption, as we studied in the biopharmaceutics. So anything less permeable because of the high molecular weight as well as the hydrogen bond or hydrophilic compound has this particular problem. So anything if you want to any new molecule if you have you want to find out what is the permeability of the molecule, then we will use to go for the Kekartu cell line system to find out the what type of the mechanism of absorption, whether it goes for the passive transport or paracellular or carrier mediated transport. By using this cell line, we can go for the which pattern or mechanism of absorption of or permeability of the molecule. As we studied biopharmaceutical classification system 1, 2, 3, 4, so we know very well BCS class 3 and 4 are has the problem of permeability. Low permeable compound will comes under the category of BCS class 3 as well as BCS class 4. Anything new molecule comes to you, if you want to find out the permeability, always we should compare with the standard product or standard molecule. So the FDA approved standard molecule is metoprolol. So whenever if you have the new molecule, if you want to compare or if you want to find out the permeability, always you have to compare with the metoprolol and we are finding the relative permeability or relative absorption, you can say that. If relative permeability less than 5%, that molecule is not recommended for oral route. If relative permeability 5 to 50, then you can able to make the formulation, oral formulation, but you have to include in that formulation absorption enhancer to increase the permeability. So by adding the absorption enhancer, then we can able to make the formulation for 5 to 50 percentage of the relative permeability. If suppose relative permeability is more than 50, that absorption is more than 50, so it will be even it is a BCS class 4. We know BCS class 4 low soluble, low permeable compound. Even though it is a low permeable, but it has a relative permeability is more than 50 percentage, the BCS class 2 will be treated as a BCS class 2 drug because permeability is more than 50 percentage. Sometimes some literature survey we used to see in some paper, some of same drug mentioned the BCS class 2 also. In some more paper, same drug they might have given BCS class 4. So sometimes we used to confuse whether it will be considered as a BCS class 2 drug or BCS class 4 drug. So because this classification according to the relative permeability they kept, that's why some people used to report as a BCS 2, some people will report as a BCS 4. Okay, even though it is a BCS 4, if it is a relative permeability is more than 50 percentage, then it will be considered as a BCS class 2 drug. Even though it is a BCS 4, but we will consider as a BCS class 2. So for low soluble, high permeable compound. So that's why we used to compare always metoprolol to find out the relative permeability. But if anything molecule shows the low permeability because of its inherent property, very difficult to increase the permeability of any molecule without making any structural modification. That's why there is no any formulation technology to improve the permeability of any molecule except one option. So only we have to give the absorption enhancer in the formulation. There is no any another option for to enhance the permeability. That's why each and every PG student and PhD scholar never work on enhancement of the permeability. Everyone will work on enhancement of the solubility, nothing but BCS class 2 as well as the BCS class 4. Now we'll discuss about solubility. You see this equation, general solubility equation, the log of saturated solubility is equal to 0.75 minus log P lipophilicity plus melting point divided by 100. So if you take any molecule, you see the what is the saturation solubility of molecule, what is the lipophilicity of the molecule, what is the melting point of the molecule. Okay, if we see these three parameter is playing important role in the solubility. Especially the solubility is mainly depends upon the two parameter. One is the log P value, nothing but lipophilicity. Another one is the melting point of the molecule. The two parameters playing important role in the solubility of any molecule. Okay, even out of these three parameter, if you know any two parameter, you can able to find out the third parameter by using this equation without doing any experiment. If you know the solubility, if you know the log P, then we can able to find out the melting point. If you know the melting point, if you know the log P, then you can able to find out what will be the saturation solubility of my molecule. So there is a interrelationship between the three parameter. One is the solubility and log P as well as the melting point. Okay, so solubility is mainly depends upon the this two parameter. You can say that. If suppose you have the molecule log P is greater than the melting point, that means log P is more, lipophilicity is more, nothing but the solubility is mainly depends upon the log P limited solubility, poor aqueous solubility. Such a molecule, how to enhance the solubility of the molecule? Only lipophilicity is more, melting point is less. So solubility is mainly depends upon the lipophilicity. So how to increase the solubility of such a molecule? Just you have to increase the wettability of the molecule, wettability of the crystals by adding the surfactant. Just you have to add the surfactant. You have to increase the wettability of the molecule, or by using the lipid solvent or co-solvent to increase the solubility of the molecule if lipo log P is greater than melting point divided by 100. If suppose log P is less, melting point is more, that means the solubility of the molecule is melting point, limited solubility, poor aqueous solubility. You can say that. That means what? Here solubility is mainly depends upon log P. Here solubility is mainly depends upon the melting point. The two parameters playing important role. Okay, so poor solubility. So why the melting point is more? See, some of the molecule having melting point is less, some of the molecule having melting point is more. What is the reason? So we know that we studied in the crystal engineering, crystallization technique or crystallization of the any molecule, pharmaceutical engineering chapter we studied. If any crystal you take, there is a two parameter. One is the crystal lattice, another one is the crystal habit. So crystal habit is the outside appearance, rod shape, cylindrical shape, square shape, and all those things. Crystal lattice means what? Arrangement of the molecule inside the crystal. How the arrangement of the molecule inside the crystal. So one molecule, another molecule, the crystal lattice bond. Okay. So inside the crystal lattice, each and every molecule is organized tightly. That means each and every molecule is bound very strong bond between the molecule inside the crystal lattice. Strong bond between the molecule in the crystal lattice. That means they have the more attractive force between the molecule inside the crystal lattice. So because of that high crystalline lattice force of the molecules, because each and every molecule is bound very tightly inside the crystal lattice, that's why it shows the more melting point. If that force is very less, loosely packed molecule in the crystal lattice, the melting point will be very less. So melting point is more mainly because of the high crystalline lattice force of the molecule, which leads to the more melting point. Automatically, high melting point always have the low solubility. So if you want to increase the solubility of high melting point molecule, then what we have to do? You have to break the crystal lattice force. So whatever the force between the molecule, you have to break the force between the molecule. So what we used to do? We used to go for the solid dispersion because the force will get break in between the molecule. Our polymer solid dispersion PVP, PVP K30, PEG, and all beta cyclodextrin, whatever the solid dispersion techniques are there, that polymer will come and bind between the two drug molecule. Automatically the force will get decreased. The force between the molecule in the crystal lattice will get decreased. Automatically solubility will get improved. So solid dispersion nanocrystal, which will reduce the crystal lattice force, automatically improve the solubility of the molecule. So here, solubility is mainly depends upon the lipophilicity and melting point. If the lipophilicity is more, you have to increase only wettability. Melting point is more, and you have to reduce only crystal lattice force to improve the solubility of the molecule. Here I put up two questions. Could we improve the bioavailability of the molecule by increasing the aqueous solubility? Is the bioavailability of this molecule is solubility limited bioavailability? The out of two question is which one is correct? Whether the first question is sometimes we used to say correct, sometimes second question also sometimes we used to say it is a correct. If any molecule shows the low bioavailability, we should not consider we have to improve the solubility of the molecule. It is totally a wrong concept. Could we improve the bioavailability of the molecule? Any molecule shows low bioavailability, directly you should not take the decision to increase the solubility of the molecule, because we should know whether there is a need of to improve the solubility which could increase the bioavailability of molecule. The first question is totally wrong. The second question you see is the bioavailability of this molecule is solubility limited. First, you should know molecule is poor bioavailability, but we should know whether there is a need to improve the solubility or not. If it is, there is need to improve the solubility, then only you have to go for solubility enhancement. Then automatically you have to improve the bioavailability of the molecule, because we should find out what is the reason for to show the low bioavailability. So how to find out whether my molecule is solubility limited, bioavailability? If it's solubility limited, then we will go for solubility enhancement and followed by improve the solubility, improve the bioavailability. So how to find out my molecule is solubility limited, bioavailability? If any molecule comes to you, any molecule comes to you, just you have to find out the absolute bioavailability of the compound molecule. What is absolute bioavailability? Oral bioavailability will be compared with the parenteral bioavailability. Just the injectable will be compared with the oral suspension. Find out the absolute bioavailability of the molecule. Whether absolute bioavailability of molecule is high? Question mark. Yes or no? Yes, that means what? Absolute bioavailability is more. That means what? The bioavailability is not solubility limited, not dissolution limited. No need to do any work on this particular molecule because the drug itself having more absolute bioavailability. So bioavailability could not be further improved. You cannot increase the bioavailability by any enabling formulation. Enabling formulation means what? Whatever we are doing, PG research, PhD research, all the formulation we are telling as an enabling formulation. There is no need to do any research on this particular molecule if the absolute bioavailability is more. If absolute bioavailability is high, no. That means what? The absolute bioavailability is very less. Then you have to go for the relative bioavailability of the molecule. What is relative bioavailability of molecule? Oral formulation will be compared with the oral. Nothing but oral solution will be compared with the oral suspension. Same dose, same drug, one in the suspension, one in the solution we are giving oral, and we are find out the bioavailability, and we are comparing the relative bioavailability of oral suspension compared with the oral solution. You can say that. Everyone knows that oral solution will give always more bioavailability as compared to the oral suspension. That everyone knows that. If suppose if we if we get the result like this, oral solution is greater bioavailability as compared to the suspension. Obviously, everyone knows solution will be always more bioavailability as compared to the suspension. That is nothing but relative bioavailability I'm talking. Yes, solution shows greater than suspension. The bioavailability of the molecule is solubility limited and as well as the dissolution limited. Such a molecule, you have to go for the solubility enhancement. The bioavailability could be improved by enabling formulation. If we get the result like this, then only we have to go for the solubility enhancement followed by improve the bioavailability of the molecule. If suppose if we get that means this result is suitable for to improve the solubility of the molecule. If suppose if we get the result like this, solution bioavailability is equivalent to the suspension bioavailability. Oral solution equal to oral suspension. Normally everyone say solution will give more bioavailable as compared to the suspension. But we get the result like this. Suppose equal bioavailability. That means the bioavailability is not solubility limited bioavailability. What is the reason for low bioavailability of the solution? Maybe there is a low bioavailability may be related to the permeability issue or first pass metabolism. This is the reason for to give the equal bioavailability. So there is no need to improve the solubility of such a molecule. You see the third result, suspension shows more bioavailability as compared to the solution. Normally knows solution should give greater than the suspension, but our relative bioavailability suspension given greater than suppose solution. That means the bioavailability is not solubility limited or dissolution limited. So what is the reason for low bioavailability? Might be stability issue in the GIT tract, maybe pH dependent unstable or chemical degradation or enzymatic degradation, something. So there is a stability issue in the GIT tract that is the contributor to the low bioavailability. So if we get the result of second and third, there is no need to improve the solubility. If we get the result this one, then only we have to improve the solubility of the molecule to improve the bioavailability of any poorly water soluble compound. There is another interrelationship between the three parameters: solubility and permeability and efficient dose to optimize the drug absorption. Efficient dose means nothing but effective dose. Okay, the interrelationship is there. Efficient animal dose, suppose 1 mg per kg, animal dose 1 mg per kg, and high permeability. Then what should be the solubility of the molecule to get the good bioavailability 10 mg per ml? If you want high permeable Animal dose, then you should have the solubility like this. Animal dose is 10 mg per kg and minimum solubility required for 100 mg per ml to get the good absorption. So there is interrelationship between these three parameters that is called as dose solubility number. Dose solubility number is equal to efficient dose divided by saturation solubility of molecule. Okay, so the relationship, if dose solubility number, what it indicate that if you get the result something, we are going to get the dose solubility number. So it indicating that how much quantity of liquid or solvent required to solubilize the efficient dose, how much solvent or how much gastric juice required to solubilize the efficient dose, which is indicating the dose solubility number, you can say that. If you get the result like this, less than 100 ml per kg, you consider this is a highly permeable compound. Dose solubility number is less than 20 ml per kg, then medium permeable compound, less than 5 ml, that is considered the poor permeable compound. So from dose solubility number, we can decide whether it's a highly soluble, highly permeable, medium permeable, or poor permeable. This is for one idea we can get from the dose solubility number. Then we can go for the later BCS classification. You can say that. Another one is another. This chart is very important. This chart is each and every industry people used to follow that. As an academician, we never follow such a flow or such a pattern to do the research. Just I want to say that any new compound comes to you. First, you see the physical chemical characteristic of the molecule, physicochemical property of the molecule. Then you find out bioavailability, solubility limited or not. Just now we discussed the relative bioavailability should be more for solubility of molecule. Just now we are discussing this slide how to find out the solubility limited bioavailability. Same here. If the molecule is bioavailability solubility limited, then we are going for solubility enhancement. If it is not solubility limited, then we are not going to do any research on that particular molecule. So bioavailability, solubility limited or not? Yes or no? If it is a no, that means what? No need to enhance the solubility because the drug itself having good solubility, and you can give as a solution form or suspension form to the patient. If it is a yes, that means what? Bioavailability, solubility limited. Yes, then you have to increase the solubility. How to increase the solubility? First you have to see the structure of the molecule, the molecule having ionizable functional group or not, COOH, NH2. Any ionizable functional group is there in the structure or not? Yes or no? If it is a yes, ionizable functional group is present in the structure, then you can convert into salt form of the molecule, like diclofenac sodium. Is it correct? CPM, chlorpheniramine malate, whatever, any salt form. Okay, ionizable functional group is there, yes, then convert into salt form to improve the solubility. If salt form is improve the solubility or not, yes or no? If it is a yes, that means salt form is improve the solubility, then you can convert into salt form and mix the remaining excipient and convert it into tablet or capsule or any suitable dosage form. No need to do any further research. If salt form is not increase the solubility, salt form also not improve the solubility, and also there is no ionizable functional group also present. No ionizable functional group and salt form also not improve. Then you have to go for the size reduction, micronization technique to improve the solubility of the molecule. Size reduction is improve the solubility or not? Yes. Size reduction is improve the solubility. Yes. Then only you have to do the size reduction, remaining mix your excipient and convert it into suitable dosage form. If size reduction also not improve the solubility, no. Micronization also not improve the solubility. Then you have to see what we discussed previously, two parameter, log P and melting point. Lipophilicity as well as the melting point you have to see. If log P value is more, melting point is less. Just now we discussed if log P is more, then how to improve the solubility? We have to do the increase the wettability of the crystal. So surfactant, lipid based formulation, surfactant we are adding to enhance the wettability of the molecule. If log P value is less, melting point is more. Crystal lattice force is more. Then we have to break the crystal lattice force and we have converted into solid dispersion. We have discussed. So you can convert it into nanoparticular size reduction, nanocrystals or solid dispersion to improve the solubility of the molecule. If suppose log P value also high and melting point also high, then what to do? You have to attempt to increase the wettability also, and you have to reduce the crystal lattice force also. That means both the solid dispersion as well as the lipid based formulation to improve the solubility of the molecule. You can say that. So normally, what will be considered as a log p value is high. What should be the level? If anything shows greater than 2 to 3 log p value or lipophilicity will be considered as a highly log p value. Melting point is more. It means anything more than 160 degrees centigrade will be considered as a highly melting point. These are all what we are given in the literature survey will be considered. What's a high log P and low log P. So this chart is very important. So this only whenever any molecule comes to you, you have to start from the that is a chart to find out the solubility of the any molecule to improve. Okay. Now we study the BCS class 1, 2, 3, 4. Okay, BCS class 1, everyone knows that. High soluble, high permeable. Nobody will work on BCS class 1 drug. Everyone will work on BCS class 2 drug. Some people may be BCS class 3 to improve the permeability by adding the some lipid based formulation. And very rarely people work on BCS class 4, improve the solubility as well as they improve the permeability. But even the BCS class 1, we should consider some of the parameters to develop the formulation. First, you find the dose of the molecule. If dose is greater than 40 mg, yes or no? Dose is greater than 40 mg, yes or no? It is a yes. Then you see the flow property of the molecule, API. It is freely flowable or not? Yes or no? If it is a no, directly go for the dry granulation method. If it is a yes, flow property problem is there. And also dose is not greater than no written. Dose is not greater than 40. It might be less than 40 mg. Admin. Then we are seeing the solubility of the molecule. What is the aqueous solubility? So aqueous solubility is greater than 0.3 mg per ml. 0.3 mg means 300 microgram per ml. Solubility is greater than 300 microgram per ml or not? Yes or no? It is no. That means what? Not greater than 3 mg per ml means it might be less than 300 mg per ml. So directly you can go for the dry blend with the surfactant to increase the wettability. Because it is a poorly water soluble compound. Anything shows less than 300 microgram per ml, then we have to go for the wettability of the molecule to improve the solubility. If it is a yes means what? It might be soluble more than 300 microgram per ml. That means 500 microgram per ml, 1 mg per ml. So directly you can go for the dry powder blend to make the suitable dosage form. This is what we studied in the BCS class 1 we should consider. You see the BCS class 2 drug, low soluble, high permeable. Then what we have to ionizable is there or not? Ionizable functional group is there or not? If yes, or then we'll convert it into salt form. The salt form will improve the solubility or not? We have to check. If salt form is not improved, then we'll go for the size reduction, micronization technique. The micronization also not improve the solubility. Then we'll go for the log P and melting point, lipophilicity and melting point to improve the solubility. This is the one direction of the BCS class 2 drug research. Now another direction, you have to see the what is the dose volume of BCS class 2 drug. So ionizable group, yes or no? Yes or no? Yes. Ionizable. Then you go for ionizable functional group or not. If the yes, then you see for dose volume of this BCS class 2 drug. What is the dose volume? Dose volume is equal to human dose divided by solubility. The human effective dose divided by saturation solubility of the molecule. At if your drug is basic drug, then you have to do the solubility in the pH 6. If your drug is acidic or neutral drug, then you have to go for the solubility in pH 4 solution. The solubility should be done in the pH 6 or pH 5. For pH 6 is the basic drug, pH 5 is the acidic and neutral drug. This is the dose volume. Nothing but the volume in which your whole effective dose will get soluble. That is called as the dose volume you can consider. Okay, so dose volume is greater than 250 ml. Dose volume greater than 250 ml. Yeah, ionizable, yes or no I written. If it is a yes, then we are seeing what is the dose volume greater than 250 ml. It means what? The effective dose will get soluble in more than 250 ml. Maybe 500 ml or maybe 600 ml. Yes or no? If it is a no, that means what? The drug might be soluble. Efficient dose might be soluble in less than 250 ml. Then it will be considered as a BCS class 1. Any effective dose soluble in less than 250 ml will be considered as a BCS class 1. If it is a yes, that means what? That effective dose might be soluble in more than 250 ml. Yes, that means 500 ml, 1 liter or 2 liter something. Yes. Then again you are saying what is the maximum dose volume? If a dose volume is less than 5000 ml, that means what? Effective dose soluble in less than 5 liter or 5000 ml. Yes or no? It is a yes. That means what? Less than 5 liter, the effective dose might have solubilized. Then you can make the formulation by adding the surfactant. Better wax granulation by adding the surfactant. If it is a no, no means what? It is not soluble in 5 liter also. It is not soluble in 5 liter also. Then what we have to do? Then there is no use to do research in the aqueous system. Now we are shifting to the non-aqueous system. Again, I'm repeating. So dose volume you have to find out if it is a greater than 250 ml, yes or no. If it is a no, that efficient dose might be soluble in less than 250 ml. Then it will be considered as a BCS class 1. If it is a yes, that means what? The efficient dose might be soluble in greater than 250. Yes, then it may be the 1 liter, 2 liter, 5 liter, whether it's a less than 5 liter, yes or no. Yes, then you can order the wettability to increase the order the surfactant. If it is a no, that means the efficient dose. So by example, paracetamol 650 mg. That means paracetamol 650 mg not soluble in 5 liter also. No. Then there is a no use to do solubility in the aqueous system. Now we are shifting to the non-aqueous system. Oily vehicle you have to find out. Non-aqueous. So then you see the solubility of such a molecule in non-aqueous or oily vehicle. You can say that sesame oil, peanut oil, sesame oil, some oil we can use to the soluble your dose. That solubility in the oil or non-aqueous system is greater than your effective dose or not. Yes or no? Saturation solubility in non-aqueous vehicle, the total solubility is greater than dose, yes or no. It is a no, then you can go this process to improve the solubility. If it is a yes, that means what? It might be greater than particular dose. Then once you make the solution in the non-aqueous vehicle, then the solution should be stable. The solution means oily solution should be stable. Yes. If it is a yes, then you can fill this solution or oily solution in the soft gelatin capsule, solution or semi-solid, filled into the soft gelatin capsule and dispensed to the patient. This is the what we used to do in the BCS class 2 drug in the research and development. So from the this much only for the what we can say high throughput screening and lead molecule. The main parameter solubility enhancement, what we are seeing the log p value as well as the melting point. What are the things should be considered BCS class 1 and BCS class 2 drug? You can say that. This much only for high throughput screening and lead molecule. Now come to the from the lead molecule, how to select the drug for formulation research. This already we studied in the GIT pH as well as the transit time. You can say that. Okay. Now you can see here. What is the pH of our GIT tract? Mouth pH almost neutral, near to the neutral, 6.8 or 6.8 around neutral. Mouth pH is the neutral. You can consider 6.8. They come to the stomach. Stomach pH, everyone knows it is acidic pH. Okay, in the stomach, top of the stomach will be highly acidic nature, and then the pH will get decreased when come down. Again, it become top of the stomach. It might be 1.2 pH. Again, while coming down, it become 2 pH, 3 pH, 4 pH, 5.5. Then when the stomach and small intestine is joining area, stomach and small intestine fundus below the fundus, you can say joining portion between the stomach and the small intestine, the pH will be 6.8. The stomach pH is acidic pH, everyone knows. Then small intestine and large intestine is alkaline pH. Okay, after 6.8, it becomes 7.2. Again, it becomes 7.4. Again, it becomes 7.2. So three pH will get exist in the small intestine and large intestine. Okay, and the colon, end of the GIT tract colon. Okay, the pH is 8. So highest pH in the GIT tract is the 8 pH. So this pH is very important. So from that pH, what we can conclude, stomach is the acidic pH and intestine and colon, everything is the alkaline pH. As basic pH you can say. So the pH of the GIT tract. Now transit time or residence time, sometimes there is a meaning is there. So what is the transit time in the mouth and small intestine, stomach and colon? Okay, if you see mouth, the grinding will be only 30 seconds or maybe 1 minute and it goes to the stomach. Any food material, you take idli, chapati or anything, if you eat, grinding only 30 seconds or 1 minute, it goes to the stomach. The chapati idli will stay around 2 to 3 hours in the stomach. 2 to 3 hours in the stomach, it will stay. After 2 to 3 hours, it goes to the small intestine. The small intestine, it will stay for 6 to 8 hours in the small intestine, 6 to 8 hours. Again, the food goes to the large intestine, again 6 to 8 hours. Then after that, it goes to the elimination, so eliminated through fecal. So if you see the transit time or travel time of the any food material, stomach is to 2 to 3 hours. You consider maximum 3 hours, and small intestine is considered as 8 hours. Large intestine considered 8 hours. So 8 plus 8, 16 plus 3, 19 hours. So after 15 to 20 hours, whatever the eating material will excreted through fecal. This is the transit time, you can say. So we know what is the pH of the GIT tract: acidic in the stomach, alkaline in the small intestine, large intestine, and the transit time also we know very well, 3 hours, as well as the 48 hours in the small intestine, again 8 hours in the large intestine. The pH, this is we know very well, we study in the school life itself. pH scale always 0 to 14. Acidic means proton donor, basic means proton acceptor, everyone knows. And again, definition of the pH is negative log of hydrogen ion concentration knows. Now, how it comes, how that pH scale comes from 0 to 14, how that equation comes, that I want to say here. Okay, any water molecule undergo dissociation, auto dissociation of water will form hydrogen ion as well as hydroxyl ion. Everyone knows this, we studied in the school itself. So hydrogen ion and hydroxyl ion will form whenever water undergo auto dissociation. That ion formation will be equal number of ions will form, hydrogen ion as well as hydroxyl ion. If we see 1 liter of the water, 1 liter of the water going for dissociation, how many number of hydrogen, how many number of hydroxyl will form? 1 into 10 to the power minus 7 ions of hydrogen, 1 into 10 to the power minus 7 ions of hydroxyl. Equal ion formation will take place when 1 liter of the water undergo dissociation. Then we can write dissociation constant. How to write? Or ion of degree of dissociation, something we used to say. The hydrogen plus hydroxyl will give 1 into 10 to the power minus 14, because minus 7 hydroxyl, minus 7 is the hydrogen. So we are summation of these two. We are writing 1 into 10 to the power minus 40 at the room temperature. You take log both the side. Forget about minus. Log of hydrogen plus log of hydroxyl is equal to that minus 14 will come here. Minus 14. Log of 1 is the 1. The value becomes 14. Minus 14. So log of hydrogen plus log of hydroxyl is equal to minus 14. I want to remove this minus 14, then multiply with both the side is the minus negative. So negative log of hydrogen, negative log of hydroxyl will give the 14. Negative log of hydrogen ion, negative log of hydroxyl will give the 14. This only we used to say pH plus POH, which give the 14. So pH plus POH will give the 14. So there is no pH scale more than 14. The highest pH is only 14 only. Then how that equation comes, that I want to say in that particular slide. Okay, so now any solution source, we know the pH scale and all those things, no need to discuss. 0 to 7 is the acidic, 7 to 14 is the alkaline. Everyone knows that, neutral. If any solutions, suppose drug dissolve in any solution, then immediately the solution shows the pH greater than 7. Drug molecule dissolved solution, any solvent converted into solution, the solution shows the pH more than 7. It means it is alkaline solution. Everyone say it is alkaline solution. pH is 7. That means what? If pH is more than 7, it means the molecule have hydroxyl ion or nitrogen ion in the molecule. Just you see the molecule structure. All heterocyclic nucleus ring. You can see here. So imidazole, pyridine, is it correct? Isoxazole, what are the quinoline, isoquinoline? These all are heterocyclic rings are having nitrogen atom, or it may contain some additionally hydroxyl ion. If the two functional group in the structure, if we dissolve the solution, definitely it become alkaline solution. The pH will be more than 7. So from structure itself, we can identify my molecule is alkaline nature or acidic nature. If pH is less than or greater less than 7, it means everyone knows the acidic solution. It may contain hydrogen ion in the structure, or carboxylic group COOH is present in the structure. From that we can conclude it is the acidic drug. So from structure itself we can conclude whether my drug is acidic drug or basic drug. Acidic drug having COOH and H, and basic drug might be having hydroxyl ion or nitrogen atom. In the structure itself we can able to identify. Okay, so in the market there is a two type of the drug available in the market. There is no drug for strong acidic drug and strong basic drug is not available in the market, because strong acid, strong base will get completely ionized in the GIT tract. As we studied in the biopharmaceutics, only unionized drug undergo absorption. Ionized drug does not go for the absorption. We study. Same here. Strong acid, strong base is not available in the market because it is completely ionized in the entire pH. What are the stomach pH to up to colon pH? It will get ionized. There is no use. That's why it is not available in the market. Only weak acidic and weak basic drugs are available in market because it is incompletely ionized. Incompletely ionized means what? Some of the parts will get ionized, some portion will not get ionized. Unionized form will get exist. That unionized form will go for the absorption. That's why only weak acid and weak bases are available in market because it is incompletely ionized in the pH range. It will exist in the unionized form undergo absorption. Already I told you how to identify my drug is the weak acid or weak base. Weak acid drug structure itself we can identify it contains COOH, carboxyl group, aspirin, acetaminophen, or whatever it may be. Okay, so it contains COOH or hydroxyl ion. We can consider it is an acidic drug. Now you can see here OH is there, two OH is there, and one COOH is there. So whether it will be considered as an acidic drug or basic drug. So now one carboxylic is equal to two OH, two molecule of OH. So here also there is a nitrogen atom is there, but even though it is considered the acidic drug because here carboxylic group is the NaCDs are derived from acetic acid derivatives. Acetic acid derivatives NaCD come from acetyl derivatives. That's why it considered the acidic drug. So one carboxylic group is equal to two nitrogen atoms. So it will be considered acetaminophen or paracetamol is the acidic drug. So from structure itself we can identify it as acidic drug. Same you see the basic drug, nitrogen should be the compulsory plus OH will be there. Now you can see here or metformin, dicyform, trimetrin or whatever any drug you take. In basic nucleus or heterocyclic containing nitrogen, it is a basic nature or basic drug you can consider. Okay, so now structurally we can identify it is my acidic drug and it is my basic drug. We know the pH of the GIT tract as well as transit time of the pH GIT tract. Now, again we'll go back to the biopharmaceutics. Okay, acidic drug in acidic pH, it will exist as an unionized form. That means acidic drug in the stomach. Acidic drug in the stomach, it will be unionized form, absorption will be good. But same acidic drug in basic pH, it will get ionized, does not go for the absorption. That means acidic drug when comes to the small intestine or large intestine, it will get ionized, does not go for the absorption. That means acidic drug absorption only in the stomach, not in the small intestine, not in the large intestine or colon. Same for the basic drug. Basic drug in basic pH, it will be unionized form. The absorption will be good. It means basic drug in the small intestine, large intestine pH is the basic pH. So basic drug in the basic pH, unionized form, absorption will be good in the small intestine, large intestine. So acidic drug absorption in the stomach, basic drug absorption in the small intestine, large intestine. So what I want to say, so what is the T max of the acidic drug? What is the T max of the basic drug? So I will go one by one parameter. All the parameter finally will play a role in the selection of the drug for oral dosage form development you can consider. Okay, so weak acid absorption stomach, weak base absorption in the small intestine. So what will be the T max of the acidic drug? So acidic drug in acidic pH unionized form, absorption good in the stomach, but the drug will stay only 3 hours. Chapati, idli or drug will stay only 3 hours. After 3 hours, it goes to the small intestine. If acidic drug goes to the small intestine, it will get ionized, it does not go for absorption. So what will be the Tmax for acidic drug? Tmax means we know the time required to get the maximum absorption. So acidic drug Tmax will be maximum 3 hours. From Tmax itself, we can conclude that my drug absorption in the stomach less than 3 hours. Same for the basic drug, you can see the what will be the Tmax. Definitely basic drug Tmax will be more than 3, because the basic drug absorbs only in the small intestine. So first 3 hours it will be remain in the stomach, but the drug should not get released. The 3 hours basic drug should travel to the stomach. We are consuming basic drug, but absorption in the small intestine, but it has to travel through the stomach. During the traveling in the stomach or 3 hours in the staying in the stomach, the drug should not get released. For that, we are giving enteric coated tablet or enteric coated formulation. You can say so. Enteric coated polymer we are giving on the tablet. The polymer is resistant to acidic pH. That means insoluble in acidic pH. That's why the basic drug enteric coated we are giving, it will stay 3 hours in the stomach, but it will not release the drug in the stomach. At like a dead body, it will stay 3 hours in the stomach. After 3 years, the enteric coated will go to the small intestine. The polymer will get dissolved because the polymer is basic sensitive polymer. It will get dissolved. The drug will get released. It is a basic drug. Our small intestine is the basic pH. Unionized form absorption will be good. So Tmax of the basic drug should be more than 3 hours, but more than 3 hours, anything will be 8 hours, 10 hours, because until to elimination up to 15 hours, 16 hours also will be considered for the basic drug. So now we can conclude this is the acidic drug, this is the basic drug, where absorption window, stomach, here absorption window is the small intestine. What will be the Tmax of the acidic drug? What will be the Tmax of the basic drug? Last parameter, just 1 minute, 1 minute. Sorry. The PK also playing important role for the absorption of the drug, playing selection of the drug. So some student never consider any physicochemical parameter. PK means what? Degree of ionization or dissociation constant we used to say. That mean if we, that mean PK is suppose 5. What is the meaning of the pH? PK is 5. That means what? At 5 pH, PK is 5. At 5 pH in the GIT tract, at 5 pH, the drug will exist 50% ionized form, 50% is the unionized form. The meaning of pKa. Okay, so acidic drug in acidic pH unionized form. So acidic drug, acidic pH 1 to 7, it will be unionized form. So when it will get ionized, when it become more than 7. When it touch to the more than 7 pH, when it come to the alkaline pH, then it will get ionized. That means acidic drug pKa will be always more than 7. Then vice versa, you can consider basic drug in basic pH unionized form. That means basic drug 7 to 14 pH, it is unionized form. When it is less than 7, it will get ionized. The basic drug will get ionized less than 7. So what will be the pK of acidic drug? More than 7. What is the pK of basic drug? Less than 7. What is the meaning of pK? At particular pH, it will exist 50% is ionized form, 50% is unionized form. Only unionized form will go for the absorption. Okay, so for acidic drug, now you can consider acidic drug pK is 8. So acidic drug in acidic pH unionized form. That means 1 to 7 pH, it will be 100% unionized form. When it touch to the pH 8, then 50-50. You can consider 50 unionized form. Again, the ionized form, here unionized form. Same you can consider for the basic drug. Here it is unionized form. In acidic pH, it will get ionized form. So where our drug going for absorption, PK is very important parameter. Where absorption window, what is the pH of that absorption window, and what is the PK of our molecule? In that particular absorption window, the particular pH, whether my drug is exist as an unionized form or ionized form, that should be considered for the selection of the drug. Okay, some molecules also 2 pKa value. You can see in the drug, what drug index or something. Okay, so we can see drug property. They used to show 2 pKa value. So it means what? That functional group having acidic functional group also, the drug molecule having structure having acidic functional group as well as alkaline functional group. That's why it shows the both pKa value, acidic also and alkaline also. That time which one we have to select? You count the number of functional group. Nitrogen, how many number of nitrogen, how many number of OH, how many number of COOH, which one is more number of is present, that pK will be considered for your maximum for the existing for absorption. So this pK is playing important role. Now you can see the aspirin. Everyone knows it is an acidic drug. Aspirin is the acidic drug. That means acidic drug in acidic pH unionized form. The pK will be always more than 7. pK will be more than 7 because 1 to 7 it will be unionized form. But even though they shows the pKa 3.5, pK that means 3.5. It means what? At 3.5 pH, it will exist 50% ionized form, 50% unionized form. So where that absorption will take place, where it will exist as an unionized form. That means pH 1, it will be unionized form. pH 2, unionized form. 2.5, unionized form. 3, unionized form. 3.4, unionized form. That means at top of the stomach, if you see the pH of top, 1, 1.5, 2, 2.53, and joining portion of the stomach and small intestine is the 6.8. That means the top of the stomach, the aspirin will exist as an unionized form. The absorption will be good in the top of the stomach. But when it touched to the 3.5 pH, 50% is ionized. When it crossed the 3.5 to 3.6, it is 100% will get ionized. That only we have given. Gastric juice, it is unionized form. Plasma ion, ionized form. Urine also ionized form because acidic drug in basic pH it will get ionized. Same for pethidine is a basic drug. In basic pH it is unionized form. When it comes to the acidic pH, gastric as well as plasma, again it will get ionized. That we should be considered for the okay. So until we studied about pKa, Tmax, absorption window, and acidic or basic drug. Last parameter is 2. Then we'll go for the exercise and the last. Now you can see here. So volume of distribution and half-life. These are the parameters should be considered for the selection of the drug. So what is the volume of distribution? The volume in which drug is distributed, everyone knows. Half-life, the time required to reduce the half of the quantity is the half-life. Everyone knows that. Okay. Now, total body water. If you see, total body weight 100 kg suppose or 100 percentage, in that 60 percentage is a liquid is present, water system. I'm not talking about blood or plasma, serum, all together 60 percentage total body water. Out of 100 percentage total body weight, 60 percentage is water is present, water system, liquid system. Out of 60, extracellular 25 percentage, intracellular, here I written separately, extracellular and here intracellular, remaining will be the what are the 30 intracellular 35 percentage, extracellular 25 percent. Intracellular inside the cell, extracellular outside the cell, you can say. Again, the 25 will be classified 21 plus 4, interstitial fluid and plasma. Interstitial fluid and plasma. That means what? If body weight is 100 kg, you have the 60 liter of the liquid. In that, only 4 liter is plasma is present. In that 4 liter. Interstitial fluid in between the cell. Intracellular 35 liter inside the cell. This is the how the water system is distributed in our body. Now you can see here, any 70 kg patient, how many liquid will be there? How much quantity of the water will be there? Out of 70, what is the 60% is 42 liter. That means what? 70 kg human having 42 liter of the water system in his body. If you take any drug orally, okay, once the drug coming to the GIT, then it goes to the absorption coming to the central compartment. Everyone studied what is central compartment. Drug is distributed to the central compartment, then it goes to the peripheral compartment, again come back to the central compartment and excreted through urination or excreting organ. So first it comes to the blood circulation or central compartment and goes to the peripheral. Okay, so 70 kg patient having 42 liter of the system water. If drug shows only 10 liter of the volume of distribution, drug shows any drug 10 liter volume of distribution, but 70 kg he has only 42 liter water, liquid, not water, liquid system. You can water liquid system, whatever it may be. So 10 liter only shows the volume of distribution. It means what? With 10 liter coming only here. That means what? After absorption, drug is remain in the central compartment, not went to the peripheral compartment, not reached to the peripheral compartment. That's why it shows the low volume of distribution, 10 liter. The human having 42 liter, but it shows only 10 liter. That means drug remain only in the central compartment, not went to the peripheral compartment. From central compartment, excreted through urination. Excreted through urination, you can check. Same, suppose 42 liter, the drug source volume of distribution is 40 liter, large volume of distribution, but he has a 42 liter. It means what? The drug is present in the central compartment also, drug went to the peripheral compartment also. That's why it shows the large volume of distribution. Drug is present in the central, goes to the peripheral, coming back to the central, then excreted through whatever the organ. Okay, so low volume of distribution means drug is remain in the central compartment. Large volume of distribution means drug is present in the central as well as the peripheral, both. So drug is present in the central. If suppose volume of distribution is 50 liter, but he has only 42 liter, but it shows the 50 liter, it means what? The drug is accumulated in the tissue and produce the toxicity. You see the digitalis, decoxin volume of distribution is 400 liter. Decoxin or digitalis volume of distribution 400 liter. Nobody having in our body 400 liter volume of distribution. Therapeutic drug monitoring TDM something is there. Okay, because it will get accumulate each and every cardiac cell and produce a cardiotoxicity something in the pharmacology. Is it correct? So it means more than what are the liters liquid is present. It means it produces the toxicity. You can consider that. Okay. So now low volume of distribution remain in the central compartment. Larger volume of distribution it is central as well as peripheral. If central compartment low volume of distribution remain in the central compartment, from that drug will get eliminated very fast because central compartment directly goes to the glomerulus kidney and excreted from the body. Half life also become less. So low volume of distribution having low half life. T off will be very low. I'm talking about only free drug. I'm not talking about the protein bound drug. Next slide we'll discuss protein bound drug. So only I'm talking about free drug. So low volume of distribution means drug will remain in the central compartment and it excreted through glomerulus urination. The fast elimination will be there automatically. Half life also become very less. So low volume of distribution, low half life. Large volume of distribution, drug is present in the central as well as peripheral. Again, it come back to the central, then excreted from the body. Half life also become more larger half life. So larger volume, larger half life. Low volume, low half life. There is the interrelationship between that. Okay. Now I'm talking about only free drug, not bound drug. Okay. Now come to the last parameter, protein binding. Now you can see here interrelationship written. No, not written. Okay, fine. So low volume of distribution, low half life, large volume of distribution, large half life. This is only for free drug. Now you can see protein binding. So protein binding, we see paracetamol and glimepiride. Paracetamol shows protein binding 20% and glimepiride shows the 90%. Why it shows more protein binding, less protein binding? Just go to the biopharmaceutics. The protein binding nothing but n number of force, van der Waals force. Okay, what you can hydrogen bond, hydrophobic bond, van der Waals force between the drug molecule and protein, or lipophilic interaction. Correct. This all the force is present between the drug and protein. The drug bind on the protein. Okay. Now you can see here 20% is protein binding, 90% is protein binding for paracetamol, glimepiride. So 20% means less amount. That means the force will be very less. Attractive force between the drug and protein will be low. So which attraction force? Just you consider log P value is low. Log P value is low means the lipophilic interaction is low. Lipophilic interaction low. That's why it shows low protein binding. Now come to the glimepiride. See the log P value is more. Lipophilic interaction will be more. Then again protein binding will be more. The larger protein binding will take place. Same time you can see for the aspirin as well as the carbamazepine also. So according to that we can put. At the same time the drug property, drug index something. Okay, drug bank. Okay, you can see hydrogen donor acceptor also we should consider whenever you go for the protein binding or whenever you go for the co-crystal. Co-crystal preparation also whenever you go to the acceptor donor is playing important role and PK also for the co-crystal preparation. So no need right now. Okay, so acceptor donor, see, two accepting, two donating. It is a sharing with each other at a same time. Log P is low. That's why it shows the 20% protein binding. Now you can see acceptor is 7. So it will accept 7 from the proteins and strong. Giving is only 3 and accepting is 7. So hydrogen bond also more. At the same time, lipophilic, hydrophobic interaction also more. 3.5. That's why it shows the more protein binding. Now, three parameters we can correlate: volume of distribution, half-life, and protein binding. We can able to triangular series we can calculate. If suppose low volume of distribution, okay, what we done? Half-life also become low. as half-life become very short half-life. Yeah, it will get excreted from. But low volume of distribution, high protein binding. Suppose low volume of distribution drug remain in the central compartment, but more protein binding. That means what? Bound drug will be more, free drug will be less. What will be the half-life? Low volume of distribution, more protein binding, the half-life will be very, very high because free drug availability is not there. When it is detached from the protein, then it becomes free drug, then it will get excreted from the body. That means low volume of distribution, high protein binding, half-life also become more. Same vice versa, you can calculate. Low volume of distribution, low protein binding, half-life also become low. Similarly, you can consider for a larger volume of distribution and all those you can consider. Okay, last two, three slides, then we go for the exercise. So log P and partition coefficient already we discussed about. So log of partition coefficient, everyone knows that. Okay, the another equation I given, log P is equal to saturation, solubility, and melting point. The same interrelated solubility equation we studied. Similarly, it is a modified equation. So if you know out of these three, any two, third parameter will be calculated. Already we discussed in the solubility parameter. Okay, melting point. The solubility will be moles per liter. Solubility unit will be moles per liter. You can use this equation and you can able to find out what is the log P of your molecule, what is the solubility of your molecule, what is the melting point of your molecule can be calculated. Everyone knows that if any molecule having high log P value, what we are conclusion? High log P means high lipophilicity. High lipophilicity means poorly water soluble compound. Immediately our decision will take place like that. If possible to have the compound having high log P value, high lipophilicity, we used to decide is a poor, but even though it is soluble in water. Why it is soluble in water? Because of the low melting point. So here solubility main contributed by the low melting point, not log P value contribution. So high log P value, even though it is soluble in water, it means it has the low melting point, less than 160 degree. Even ibuprofen you take melting point is 75 degrees centigrade, it is soluble. But even though it comes under BCS class 2 drug, later we can discuss that. Okay, soluble low melting point. Similarly, low log P value. Low log P value means what? Lipophilicity is less, hydrophilic will be more, that more water soluble. Even though low log P value compound, still it is insoluble in water. Still it is insoluble in water because it might be having high melting point. The reason for insoluble or reason for low soluble because of the high melting point, high crystal lattice force contributing to the less soluble in compound. This is a relationship we can consider that. Okay, these are what should be the log P value for minimum to penetrate the CNS, oral, intestine, colon, sublingual, percutaneous. What should be the formation doses for injectable, oral, transdermal? What should be the log P value should be that I have given. So what should be the minimum value for absorption? You can say. Everyone knows that BCS class 1, 2, 3. According to the solubility, permeability, we have classified. BCS class 1, 2, 3. Okay, high soluble, high permeable 1, and 2 is the low soluble, high permeable. You can say, everyone knows that. Now you can see the x-axis and y-axis scale. You see, okay, x-axis scale. Already I told you the effective dose soluble in less than 250 ml. Effective dose soluble in less than 250 ml, it will be considered as a BCS class 1 highly soluble compound. Effective dose means paracetamol 650 should get soluble in less than 250 ml. That's level given here. Solubility less than 250 ml effective dose soluble will be considered as a highly soluble compound. If it is not soluble effective dose within 250 ml, it is a poorly soluble compound. That's why we have considered 250 ml, because whenever you consume any tablet, we should take 1 glass of water. 1 glass of water contain 250 ml of the capacity. Is it correct? So that's why if your effective dose should get soluble in 250 ml, the consider as a highly soluble compound. If it is not, it is a poorly soluble. This is one parameter. Another parameter you can see here, x-axis you see. The solubility is zero point 0.1 mg per ml, nothing but 100 microgram per ml. Any molecule shows greater than 100 microgram per ml, it is considered as a highly soluble compound. Okay, you are telling always BCS class 1 means high soluble, high permeable. What is the high means what? What is the level? There should be some target level. High soluble means what is the meaning of high soluble? So any compound shows more than 100 microgram per ml, solubility will be considered as a highly soluble compound, class 1 and class 3. If anything compound shows less than 100 microgram per ml, it will be considered as a poorly soluble compound or low solubility soluble compound BCS class 2 and 4. Same for the permeability. Any compound shows more than 90% of the permeability is considered highly permeable class 1 and class 2. Less than 30, sorry, less than 90, less than 90 will be high soluble, low permeable compound is class 4 and class 3. So that everyone, this is a very important. The limit is very important. Okay. So 250 ml is the another concept, and 100 microgram is a two concept can be used for solubility of the molecule. High soluble, low soluble. We can able to classify. If class 2 low soluble, high permeable, then these are the possibility to improve the solubility, pH assessment, solid dispersion, again smells, salt form, polymeric moisture, then converted into class 1 formulation. Similarly, for class 3, you have to increase the permeability enhancement by using this technique converted into BCS class 1 formulation. For 4, we are using the both atom to convert it into that. The solubility already we discussed. Okay, pH dependent solubility means see. According to US FDA, what is highly permeable? Any compound shows 90%. Here we written the scale you can see 90% permeability greater than that will be considered highly permeable. That is according to the US FDA. High permeability means what? It should have the more than 90% of the human permeability. That will be considered highly permeable. According to the WHO, highly permeable will be considered as more than 85%. According to the European Medical Association, what is high permeable? 100% it should show the permeable. Then only it is considered highly permeable. According to the different regulatory agency consider. pH dependent solubility always we have to, no need to go for all the pH dependent solubility. Only you can go 1.2, 4.10, 4.8 you can go, sir. So dissolution, this everyone knows. Immediate release tablet means what? That effect, what are the 80%, 85% of the drug should get released in the 30 minutes will be considered as immediate release. So remaining you people knows very well. These are the some of the marketed product in USA. They done one survey. They collected 185 drug from the market. They done the solubility. Then they classify the BCS class 1, 2, 3, 4. Now you can see BCS class 1 high soluble, high permeable. Out of 185, 20 percentage comes under the BCS class 1 high soluble, high permeable. Even though you see the log P value is 5. Log P value 5 also present in the BCS class 1. Log P value 5 high lipophilic. That means low soluble, everyone used to say. But even though it comes under the criteria BCS class 1 because melting point is might be low, 45 degrees centigrade. Low melting point is contributing to the solubility. Like this, you can see this chart. If we need this PPT, I will give to the FDP coordinator. You can read on that. Okay. So this is up to this we are studying biopharmaceutical. Now, in future, after 10 or 20 years, the BCS classification will get closed. There is a new classification going to come into the market, developability classification system. We used to say DCS classification. So now we study the BCS class. Now going to convert into DCS class, developability classification system. So first I will tell you what is the meaning of this between these two. The BCS class solubility will be checked in the buffer solution 6.8. Dose will be well defined. Paracetamol 650 mg. That should get soluble in 250 ml. Then only soluble in water. Correct? Solubility checked in 6.8. Dose is well defined and 250 ml is soluble. The classification 4 classification. BCS class 1, 2, 3, 4. Only 4 classification is there. And permeability cutoff 85% will be considered highly permeable. We studied about chart. That US FDA is the 90%, WHO is the 85%, and European Medical Association 100%. So we are always comparing the absolute availability in the BCS class. But in BCS class, never talk about particle size. Whether particle size is playing important role in the BCS class. Never talk about that. At the same time, dissolution also we never talk in the BCS classification. Now, in the DCS classification, we are going to talk about particle size as well as the dissolution rate. Because particle size and dissolution rate is interrelation. Less particle size, what will be the dissolution rate? More particle size, what will be the dissolution? That will be considered in the developability classification system. This is the difference between this and that. Okay. So here, the solubility, what is the DCS? The buffer we done. Here we are going to do the, in vivo mimicking. Jejunal solubility we are going to do. So biorelevant solubility, we are not going to use buffer 6.8. We are going to use biorelevant solubility. Okay, faster state simulated intestinal fluid, FASSIP. Solubility will be checked in the as like a in vivo condition. what our regional pH. Okay, dose is not depend what are the dose, but the solubility here 250 ml here going to convert into 500 ml. Any drug is soluble in 500, it will be considered the class 1 high soluble compound. Classification here 4, but here 5. The second classification will be divided into second A and 2B. That means DCS2A and DCS2B. But here we studied only BCS2. There is no A and B. Later we'll discuss. So additional one classification. Again, the permeability will be human. It should get mimic to the human permeability. Human permeability will be 1 into 10 to the power minus 4. Then only consider. Okay. Here we never consider particle size. Same only absolute point. Cell line study will be considered for permeability. Particle size is playing. Solubility is together. 3 dose minimum particle size before absorption become dissolution rate limited. So here we are going to consider the solubility and dissolution rate. Now you can see that DCS classification, how the 2A and 2B. Now DCS 1, this is the 2, okay, this is the 3 and this is the 4. 1 we know high soluble, high permeable. 3 no, soluble, poorly permeable. 4 we know, poor soluble, poor permeable. DCS, developability classification. But in the scale, we made it 500 ml. What is 500 ml? Nothing but solubility limited absorbable dose, SLAD. But in BCS class, we used to study maximum absorbable dose. That is the maximum dose in 250 ml. Maximum dose in the 250 ml. Here, no, we are not going to talk about dose here. Solubility limited absorption dose only we are going to make. Here 2A. This is the 2. This is the 2A and 2B. 2 means what? Low soluble, high permeable. Everyone knows. Low soluble, low permeable. Again, the 2A and it is a 2B. What 2A is going to say? It is a poorly soluble, high permeable, but the solubility is dissolution limited solubility. 2A. 2B, the solubility is solubility limited solubility. That means what? Any drugs comes under the 2B, you have to increase the solubility. That means solid dispersion, lipid-based polymer, beta-cyclodextrin complex, all the enabling formulation you have to do in the 2B because solubility limited bioavailability. If it is 2A, no need to increase the solubility. So what you have to increase only dissolution rate. No need to increase the solubility. Only you have to increase the dissolution rate. How to increase the dissolution rate? Only size reduction. Particle size reduction is sufficient. Directly you can go for the formulation development. So that only we have made the classification. Now you can see here how it is converted into 250 ml to 500. 250 ml is the BCS class. 1, 2, 3, 4. Now 2A, 2B. 2A is the dissolution rate limited. 2B is the solubility limited. Here we have to increase the dissolution rate. Here we have to increase the solubility rate. That's all. Okay. Now you can see the 250 ml for BCS class, poorly water soluble you can consider, but here 500 ml. Okay, because some of the drug might be soluble in 400 here, suppose in between. If it is a 400, then according to the BCS it is a 2, but according to the DCS it is a 1. DCS 1. That only we are getting. So if any compound comes this category, only you have to increase the size reduction, increase the dissolution rate. When it comes to the 2B, enabling formulation, nothing but lipid-based formulation, amorphous solid dispersion to improve the solubility, we have to go for. The mefenamic acid comes under 2A, not 2B. 2A, mefenamic acid, we done lot of solid dispersion. N number of paper has been published to improve the solubility by PVP, K30, beta-cyclodextrin, polyethylene glycol, some of the porous polymer and all those things. But no need to improve the solubility of mefenamic acid. Only you have to reduce the particle size because it comes under the 2A according to the developability classification. So bioavailability known to be highly particle size and dissolution rate dependent. No need to improve the solubility. Same, this is the formula. What should be the size which is needed for the what are the 2A and 2B? That I have given this formula. Same again, grisoflavin comes under the 2B. So here you have to improve the solubility. But in our biopharmaceutical book, they have given grisoflavin is something you have to micronization will improve the size reduction, will improve the solubility. Actually, exactly according to here, it is a 2B, you have to improve the solubility. And again, you can consider decoxin, which come under the which category? 3, size reduction. So now it is a comparison. Now you can see ibuprofen. Ibuprofen solubility is more than 250. More than 250. It means it maybe comes under the BCS class 2 because it is greater than 2. But when it comes to the DCS, it comes under the DCS class 1 highly soluble compound. So some of the compound is shifted from BCS2 to DCS1 also. So these are the area, mefenamic acid, this all we have to do only size reduction. Here we have to do improve the solubility. This much only. This is interrelationship between the solubility and log P value. What are the formulation can be possible? Suppose log P value is 9. These are the formulation is possible. Lipid based formula. Log P value is 1. So what you have to bulk crystal, microcrystal, nanocrystal. Log P value is 3, 4, 5. Then you have to go for amorphous dispersion according to the solubility I written. Then you can refer that. I think this I now need to discuss about drug delivery. Everyone knows what is immediate release and modified release. And modified release will be classified as a delayed release, extended release, targeted release. Again, extended will be sustained release and control release. So this everyone knows that, so no need to discuss. Now come to the exercise. So audience can give answer for this. Now you can see ketoprofen. So whether it is acidic drug or basic drug, audience can open and you can suggest the what is the nature of the molecule, what type of the formulation is possible. Audience can say ketoprofen, it is an acidic drug or basic drug. Now you can see here, ketoprofen is an acidic drug because of COOH. So it is an acidic drug. So acidic drug, what will be the absorption window? The acidic drug in acidic pH, that means stomach is the absorption window. So it is acidic drug, the absorption in the stomach. Okay. So what will be the Tmax? Tmax will be less than 3. Now you can see here, Tmax is less than 3, 0.22, 0.5 to 2 hours. Yes, Tmax is only 2 hours. That means Tmax from structure itself we can identify acidic drug, absorption window, Tmax. Now you see the pK value. Now pK is 4. pK is 4. So where that 4, 3.8 means just to consider as a 4. Where the 4 pH will get exist in the stomach. Where the 4 pH will exist in the middle of the stomach. Top of the stomach 1.2, 2, 3 like this will come. So it will exist below the or above the top bottom of the stomach. That means at 3.5, at 1 pH it is unionized form, 2 pH unionized form, 3 pH unionized form. When it comes to the 4, it will get started the 50-50 ionized. That means what? Top of the stomach it will be unionized form. That means what? This drug is suitable for, the drug will exist only top of the stomach is an unionized form. That means the drug is suitable for floating drug delivery system. The drug is suitable for floating drug delivery system. Now solubility, so poorly water soluble compound. Okay, then you have to increase the solubility also. The log P value already so highly soluble. Lipophilicity. So and also you have to improve the solubility. Only we can go for half life. We see here 1 to 1.5. Half life also playing important role for immediate release and sustained release. Half life is more, then we used to go for immediate release. Half life is less, then we can go for the what we can say sustained release or control release. Now half life is very low, then we what this drug is suitable for floating sustained release. Floating sustained release is suitable for this particular molecule. Same for acetaminophen, because it's considered as a paracetamol, it is an acidic drug. Even though it is a amino functional group is there, but even the carbo is the acetyl derivatives. Okay, acetaminophen is acidic drug, then it absorption in the stomach. So what will be the Tmax? Less than 3 hours? Yes, Tmax is 20 minutes. That means then you can come to the so acidic drug absorption in the stomach and Tmax is less than 3 hours. Yes, 20 minutes. And pK. Now you see the pK. pK is 9. There is no stomach pH is 9. That means entire stomach pH it will exist as a unionized form. Even in the small intestine also, large intestine also, it will exist as a unionized form because the highest pH in the GIT tract is 8. Colon pH is the 8. So you can make entire GIT will be exist as an unionized form. Absorption will be good. Whether you need sustained release or control release, see the half life. 4 hours. You can make sustained release also. You can make immediate release also. According to the aim of your project, now you can go further. Now you can see the pentaprozole. Pentaprozole, see the structure. It is a nitrogen atom. Basic drug. So basic drug absorption will be in the small intestine, large intestine. So what will be the Tmax? Tmax will be more than 3 and more than 3. What 5, 6, 7, 8, 10. Okay, that means basic drug. Definitely it need enteric coated tablet, enteric coated formulation, because the drug should not release in the stomach. So enteric coated tablet you can make for this. Next one is whether enteric coated you want immediate release or sustained release. Then you see the half life. Half life is very short. That means you have to make, so T max is more than 3. Okay, elimination is 1 hour. Half life is 1 hour. Then you have to make enteric coated sustained release. You have to make metformin hydrochloride. You can see metformin available in market as a glycomet SR tablet, sustained release tablet. Metformin. If you see the structure of metformin, it is amino group is there. It is a basic drug. Metformin absorption will be in the small intestine because of the basic drug. T max will be more than 3. Okay, then definitely metformin should have the enteric coated tablet because the drug should exist only in the intestine, not in the stomach. So metformin SR tablet available in market. It is an enteric coated tablet because of it is a basic drug, absorption in the small intestine. Enteric coated is available. Now enteric coated immediate release or sustained release. You see the half life of the metformin. It may be short half life. That's why they made enteric coated immediate release or maybe immediate coated sustained release they made metformin hydrochloride. Even though we are taking before food, I think because it has to go fast to the after food. If you take it take longer time in the stomach. If it stomach followed by food, it will push the drug and goes to the small intestine very fast. They used to say always like that. So this is a pentaprosal enteric coated sustained release. You can say the carbamazine mean also same exercise. Is the amino group is the basic drug, so absorption in the small intestine. Okay, it must need enteric coated tablet, and Tmax will be more than 3. Now Tmax is more than 3 will be there. Now enteric coated immediate release or sustained release is the half life. Half life is 25 hours, 25 to 65 hours. So no need of sustained release and control release. Now you can make enteric coated immediate release. You can make. Now last slide. Synarizine and flubendoxil. Now you can see Synarizine, you can see the molecule structure is the basic drug because of the nitrogen atom. Because of the nitrogen atom, you can say it's a basic drug. Yes, the absorption window will be the small intestine. The Tmax will be more than 3. Yes, the given or not. See, Tmax will be more than 3. Yes, absorption is the small intestine. Then it must need enteric coated tablet. Correct? It must enteric coated tablet. Whether enteric coated immediate release or sustained release. See, the half life is 23 hours. You need enteric coated immediate release. Yes, Synarizine need enteric coated immediate release. Now you can see here another one drawback of this molecule: metabolism, hepatic first pass metabolism. Even though according to the property it need enteric coated immediate release, agreed, 100% correct. But it under the hepatic must pass metabolism, it will be inactivated. Whatever the drug is absorbed, it become inactive. Be careful, so you can consider all the parameter whatever we discuss. You can consider because of hepatic first pass metabolism, no use to give enteric coated immediate release tablet. Then what we have to do? We get basic drug. So what we have to do? Now you see the dose. Such a condition you have to see the dose. Only 25 mg tablet. So such a time, such a property, then you see the dose. Dose should be less. Then you can make the mouth dissolving tablet. Mouth dissolving tablet. Buccal batches, sublingual tablet, mouth dissolving film, because mouth dissolving film will make up to maximum 50 mg dose maximum. So you can make mouth dissolving tablet because absorbed from buccal cavity or mouth, it does not reach to the liver. It reaches to the directly to the systemic circulation. It will bypass the first pass metabolism will not takes place. So such a condition, you have to make only mouth dissolving tablet, the film, everything you can make. Same for the second drug also you can see here. So if you see the structure, it is a basic drug. NN is the August basic drug. So absorption in the small intestine, you need it. Enteric coated tablet. You see the half life, yes, 39 hours. So enteric coated immediate release we need for the flubendoxil. Enteric coated immediate release. Now you can see that inactivation of molecule, hepatic first pass metabolism as well as gut wall metabolism. The molecule is inactivated in the GIT itself. So no use to make enteric coated immediate release. Then you see the dose, just to 0.5 mg, 1 mg dose. So 1 mg dose, then you can make for the, because in buccal it is almost neutral. The drug will exist as an unionized form. You can see the pKa. So pKa is of 8.4. pK is 8.4. Our mouth pH is not 8.4, 6.8. It will be exist as an unionized form. Then you can make mouth dissolving film. and mouth dissolving tablet, sublingual, whatever want, you can make. If half life is short, then mouth dissolving film we cannot make. We have buccal batches, sustained is mucoadhesive tablet, mucoadhesive buccal batches we can make for sustained release when half life is less. Like this we can make. Tudor we can refer. So oral, sublingual, transdermal, injectable. What is the maximum dose allowed for that? This is the transdermal drug delivery system. So whatever we discuss, selection of drug for oral formulation development. Again, there is a separate lecture you can take for transdermal drug delivery. This is available in each and every book, Jane book, and all those. You can refer that. Oral dispersible tablet. Already I told how the property of the oral dispersible tablet. Okay, these are the another in different place. If anybody need such a particular topic, always welcome. Always I can deliver. Selection of drug for formulation research for PG and PhD scholar. How to write and publish effective research paper. Basic pharmacokinetic. The basic pharmacokinetics will deal about whenever we go for the bioavailability studies in animal, preclinical study, kinetic study, we used to make Tmax, Emax, AUC calculation by using the software. We know in 4 software or Kinetica. But in this lecture, without using software, we can able to calculate all the parameters. Nanotechnology for formulation development, nanomedicine in the targeted drug delivery, how to write the effective research grant, and what is meaning of journal metrics. These are my different area I used to deliver the lecture. Thank you. If you have any questions, you can ask me. Thank you, sir.
Okay. Thank you, sir. It was indeed. Thank you, sir. It was indeed a great session. Ladies and gentlemen, as we draw this enlightening session to a close, I would like to take a moment to extend our deepest gratitude to Dr. Bothiraja, sir, for his exceptional presentation on the topic how to select drug molecule for formulation research. Dr. Bothiraja's expertise and comprehensive insights into the selection of drug molecules have been incredibly valuable to all of us. The knowledge shared today will undoubtedly have a lasting impact on our research and understanding of formulation science. I now humbly request Dr. Bothiraja sir to accept the certificate as a token of appreciation. Stop sharing. I thank you again, sir, for taking time out of your busy schedule and sharing your wealth of experience and wisdom with us. We're truly grateful for the effort and dedication you have shown throughout this session, and we hope to see you join for more such engaging sessions in the future. We hope the information shared today by sir will inspire and guide all our attendees in their future research endeavors. Thank you once again. Thank you, sir. Third session, sir.